One of the questions that I'm very often asked, maybe not as much as diet and exercise and lifestyle, is the stem cell. Even this morning, since this morning, at least 10 people have asked me, how about stem cell research? And we have Dr. David Gamm, who is the director of McPherson Eye Institute and associate professor, who's probably the nationally and internationally known in his work on stem cell research. So we have the best. David?
Thank you, Suresh. Well, thank you, Suresh. I never get tired of hearing that introduction. I don't know if it's true, but I don't get tired of it. As Suresh mentioned, I've been working in the field of stem cell biology since, about 2004 or so, when I started on faculty. And it's a really unique environment here in Madison to be working in that field. And part of the reason why I think we've done as much as we have in a short period of time here at Wisconsin is because this really is the birthplace of stem cells in many respects. Jamie Thompson was the kind of the inventor of embryonic stem cells, was the co-inventor later on of induced pluripotent stem cells, which is what I'm going to talk to you mostly about today. Induced pluripotent stem cells are adult-derived stem cells. They're taken either from a small piece of skin, or more recently we tend to use blood samples like you might submit for a cholesterol check. And we can use those small blood samples, spin down certain cells that are in the blood, and then genetically reprogram them back to a very primitive state where the cells can become any cell type in your body. And that's a great thing because it gives you a lot of flexibility, and it gives you a source of cells, human cells, your own cells, that otherwise we can't get a hold of. There are certain parts of your body we can't biopsy, and certain parts of your body that don't regenerate. Your brain being one, retina being one, because the retina really is a part of the brain. I think that's important to remember in all of this, is that the retina, which lines the back of your eye, and you've heard a lot about today and probably already know a lot about, is born from the front part of the eye. So it's very similar to the frontal cortex. And just like we don't have ways of regenerating neurons in the brain when we have Alzheimer's or Parkinson's or those sort of things, same thing with regard to the eye, and in particular the retina. So for the longest time, we're kind of stuck, because if these cells degenerate, whether it be in the brain or in the retina, there's no replacement parts out there. So your options are to just get along with it, or try to make other cell types work a little bit harder, or bypass the whole system by putting in, more recently, prosthetics and things like that. So what really stem cells did when it came onto the scene, especially pluripotent stem cells, and I should say that there are a lot of things that are called stem cells. There are stem cells in your bone marrow, but they make blood, and that's all they can make. And so even though people are taking bone marrow stem cells and putting them in the eye at times, that's really just to try to make the eye happier. That's not to replace cells or to restore cells that have been lost in the course of disease. So it's important to remember that. People throw the word stem cells around an awful lot, especially on the internet. You've got to be very careful about anything you read on the internet, especially about stem cells. My laboratory is at the Weissman Center on campus, and I'm very fortunate to be there because I'm surrounded by folks who work on neural stem cells too. So they're interested in using this technology to help folks who have degenerating cells in the brain, for instance, Parkinson's and Alzheimer's and Lou Gehrig's disease, or in the spinal cord. And so we work together. I work in the retina, they work in other parts, to try and pool our resources and expertise to try and find a way to replace these very sensitive, very fragile cells that are in those parts of our body. Okay? So my mission of my lab then is to use this stem cell technology. We developed it in large respect over the course of a number of years, and now that we've developed it and we can make the spare parts that we need, can we get them to integrate and restore vision? And ultimately what we want to do is not just the laboratory work, but we want to translate what we do to help patients. And my laboratory in particular focuses on two areas, macular degeneration being one, and retinitis pigmentosa, which is an early onset degenerative disease of the retina that affects kids and teenagers, and sometimes young adults are the first to know that they've got the problem. So I'm interested in them as well. I'm a pediatric ophthalmologist, and so I see mostly kids in my clinic, and as you can imagine, while it's very, very difficult to get the news that you're losing your vision or find out you're losing your central vision, it's awfully hard to get the news that your child is going to go blind in a few years, and there's nothing you can do about it. And I think like all physicians, we don't like that helpless feeling. I think we're second in line from the patient and perhaps the family in terms of being ticked off when we get diagnoses that we just got to live with. And so having the research arm of what I do really helps not only in educating my patients, but also in feeling as though we're really making a difference moving forward and not just accepting our fates. So those are the two types of diseases I'm showing up on the bottom of this particular slide. The normal retina is shown on the left. It's a nice, healthy retina. The retina in the middle is a patient who has diabetic retinopathy, and the one on the far left shows all the scarring and changes in the center of the retina that contribute to macular degeneration. And you've heard a lot about that already today. And so a person with a perfectly normal retina would see these two children shown there completely normally. Somebody with diabetic retinopathy would have spotty vision loss and ultimately can go blind from that. And somebody with macular degeneration, as this audience is all too familiar with, loses that central portion of their vision, which is the most important part, especially as you get older and you want to read and see faces and travel. So you've gotten a lot of probably anatomy already, so I'm going to go through this pretty quick. But if we take an eye and we cut it in half, which is fun, and look at it from the side view, there's a big hollow space, and the retina lines the back of the eye like a transparent or a cellophane-like wallpaper. And if we take just a piece of that very thin tissue and blow it up and look at it under a microscope, it's not just a bland little transparent sheet of something. It's actually a very elegant layer cake. It's got a bunch of different cells that are all precisely arranged. At the very, very bottom is the cells called the retinal pigment epithelium, and you've heard about that, or RPE. And that's a very critical cell in macular degeneration. And they're the handmaidens of the retina, okay? So their job is, they exist entirely. This is what they kind of look like. If you look at them on face, so if you kind of peel away the retina and look at the RPE from the top view, they look like these little brown stop signs, all packed together against one another. And what they do is that they serve the photoreceptors that overlie them. So the photoreceptors are precisely arranged. They interdigitate with the RPE. And the photoreceptors are hard-working cells, but they're extremely needy. They can't take care of themselves. So they still live at home. They still need their mom. And so when they get rid of the various waste products and garbage, the RPE picks up after them, makes their bed, and then gets rid of everything out the other side. Okay? So the RPE has a very important role. It keeps the photoreceptors happy. And the photoreceptors are pound for pound the most active cells in your entire body. So they require a lot of help. And they're connected via this kind of two-wire arc back to the brain. So it's relatively simple. So even though it's part of the brain, it's simpler than the brain by far, at least from a logistics standpoint. And that's why the eye has led the way in terms of transplantation and thoughts about moving forward towards stem cell replacement of cells, because it's theoretically and technically easier to do in the eye than it would be to do in the brain, and safer. All right? And there are other cell types that are in the retina too that kind of mediate that signal that the photoreceptors send back to the brain to help you see. So light passes through the entire retina. That's why it has to be transparent. It sends a signal back to the brain, and that's where you actually see. You know, your eyes send signals, your brain does the seeing. So in macular degeneration, you have a slow decay of that retinal pigment epithelium, that supporting cell layer, with a lot of other changes that I won't get into. And secondarily, in the absence of its mother, the photoreceptors die. And that's when you lose vision. So ultimately, you're left with a situation where you're missing a couple of key parts that are required to see. And over time, even the other cells that are downstream of that will eventually say, well, we've got nothing to do. And so they come to decay as well. So over time, the problem gets more and more complicated. All right? And sometimes in macular degeneration, you also can have bleeding, and that will accelerate the whole process. And that's the wet type. So stem cells. So stem cells have how do they come in? Well, they're the source. They're the potential source of the spare parts that we might need to hopefully rewire the retina. Now, it sounds awfully simple and easy, and it's not. But the first step is to be able to actually make the cell types and be sure that they're what we really want, because these are very complex cells. So stem cells have a couple of basic properties. They can divide, make more of themselves. So we have an indefinite supply. We can make them from any patient and expand them indefinitely. So we have an unlimited supply of any cell type that we need. And then the trick is to induce them then not to become heart or lung or cartilage, but just the eye. And that's tough, right? Because these cells are very versatile, but at the same time, that could be a bad thing, because if they want to make everything in the world, you can't stick everything in the world into the eye. So you have to be able to harness that power and get them to make just the cell types that you're interested in. And if you can do that, then what I'm showing in this particular cartoon is we can take a patient who has a particular disease of the retina, take a sample of their blood or their skin, convert that to this very primitive state, this induced pluripotent stem cell, this adult stem cell state, and then we can do a couple of things with it. At that point, the cells are from the patient, and they still have all the same genetic and background and all the little quirks and subtleties that each one of us have, all programmed in them. So if we can take those cells and turn them into retina, essentially like recapitulating that patient's retina in a dish, so we have lots and lots of it, we can study it. And a lot of the diseases that we're interested in, including AMD, we have very relatively poor understanding about how the cells actually die in those diseases. So if we can take actual patients' cells and study them, we can learn more about the disease and hopefully develop a more educated approach to treatments. So even if we don't put the cells back into the patient, we still have this great source of cells to be able to study, learn more about the disease, and then use those cells to screen drugs, perhaps gene therapies, and other types of more conventional therapies that can get to the patient faster than a transplant. But if we do want to eventually get those cells back into a patient, then it might be that we have to fix them a bit, right? If those cells went bad, especially in genetic diseases like retinitis pigmentosa, we've got to be able to take the bad genes that led to the disease out and put good copies in. And that's one of the things we've been able to do in the past few years. And then hopefully get those back into the patient. So what I'm going to talk about briefly is just a little bit of the science behind how we can take those stem cells and turn them into something as really elegant and specific as a retinal cell. And it goes back to development. So what we try and do is take those primitive cells and usher them through step by step what they would go through if they were developing in a tiny embryo inside the mom. And so in the red circle there, what I'm circling is the very earliest part of your retina. The retina starts out as just these little tube balloons that pouch out from the growing brain. And they look like, kind of like a hollow mushroom on a stalk. And so what we were able to do, and I won't bother you with the details, but it took a number of years, was maneuver the cells through through a series of steps, both mechanically manipulating them and adding various factors. And we could get those little mushrooms, and we could get them to grow in a dish, and we could get them to separate out away from all the other contaminating cell types. So essentially then, we can get all those tiny little early primitive retinas, pull them out, and then watch them grow. And as they grow, they'll make all the different cell types of the retina in the precise order, and sometimes if you treat them just right, they'll actually layer out in the appropriate way. So the right cell will be on this side, and then the photoreceptors will be on this side, and they'll connect, which is really kind of remarkable. And we don't have to do a whole lot. We just kind of have to get out of the way, provide them with a few goodies, and they'll do it on their own. So ultimately then, we were able to coerce the whole system to be able to purify out the different cell types of the retina, including the retinal pigment epithelium, which took a little bit more finagling, but we can take those same little primitive retinas and treat them a certain way, and then they'll produce that very pigmented, dark retinal pigment epithelium. Okay, so that's great. We can make the different cell types, but how do we then intend to use that technology to help patients down the line? And so the first thing I'm going to show you is a couple of examples. One would be how to use this technology without putting cells back into a patient, okay, which takes a long time. The first trial for at least induced pluripotent stem cells to go into a patient's eye for retinal disease just started a couple weeks ago in Japan. They have one patient that they did, but it's going, and we're kind of coming up on that as well. But before we get to that point, which takes a lot of hurdles, and there's a lot of regulation, a lot of safety you have to show, and that's important. There's things that we can do to help patients right now that do not require transplants back into them. So this is an example of a couple of patients that have a disease that's like macular degeneration. It's caused by a gene defect instead of just normal aging with environmental effects. And it's called Best disease. So Best disease is a form of macular degeneration. It occurs earlier in life. And so I was interested in this disease, and so I got a hold of a couple of very, very nice families from the Illinois area, and we created, and who had this disease, and you can see it on the top, if you can see that, the central area is a little bit schmutzy. So it's not normal, and they lose central vision, just like patients with age-related macular degeneration do. So we took samples of their skin and produced induced pluripotent stem cells from them at the Weissman Center. That's a picture of the Weissman Center at the UW. made the appropriate cells, and then used the technology I just briefly went through to make the cell types that are targeted and that die in that particular disease. In this case, retinal pigment epithelium. It's the same cell type that is primarily the target for age-related macular degeneration too. So we have an unlimited supply. We can make this patient's retinal pigment epithelium up and down the door, which we do. And this is a picture of one of the patients holding one of our dishes filled with his own retinal pigment epithelial cells. So what he's holding is probably 100 times more than he has in his own eye, okay? But we're not putting those back into his eye. We can't. That's done in a laboratory, and it's not done in a clean room facility. But what can we do with it? Well, what we don't know is how that particular disease actually caused him to go blind. We don't know that in any of the patients, really. There's speculation, but we don't know too well. So what we could do is we could study it. So, and we fed it lots and lots of things that normally the retinal pigment epithelium has to ingest, and all the garbage that it normally has to deal with from the photoreceptors. And on the right side of this of the screen, the two little green, all the little green squares you see, each one of those is an individual RPE cell. And we fed them a lot of garbage. And the two on the left are from the siblings of these patients who have no disease. And those cells were able to gobble up the stuff they needed to and get rid of it. They didn't hang on to it. Whereas on the right, all those little red dots are retained garbage that the cells could not handle. And so they built it up over time. Okay, great. So we're able to see a little bit of what happens in that disease in a dish. So what can we do with that? Well, if we can understand why those cells can't eliminate the garbage they should, maybe we can find some drugs that are already available out there that can help the cells do that job for them. All right? So can we goose the system a little bit for the patients that can't do it on their own? And so we used a drug screening approach. So what we found was a couple of different drugs that were able to take those little red dots, all that garbage, were able to eliminate them over a period of time. And so the nice part about it is the drugs that we found are relatively safe. They're used by many, many individuals for their lifetime. And so if we identify the folks early enough with this disease, we can treat them with that and hopefully slow down or perhaps halt the progression of the disease. And we couldn't do that with patients, but we can, and there's no mouse model or other animals that we can use if we wanted to do that. But instead, we can actually test that patient's cells and make sure that drug works for him. All right? So that's something that we can do right now to help patients with this technology, short of actual transplantation. So to finish then, I just want to mention a couple of things, get you up to speed on the transplant end of things as well. So while what I just talked about is very promising and it's become very mainstream now, a lot of big pharmaceutical companies are talking with me and other laboratories that do this because they want to get a hold of these high throughput cell types, ways to make lots and lots of these cells so they can test drugs really quickly and use that to lower the price of drugs down the line. Not just eye cells, but cardiomyocytes or pancreatic cells. They want to get a whole battery of different cell types and use those to screen or create new drugs. And so we're definitely going to see the impact of stem cell technology on just the normal production of pharmaceuticals in the very near future. But in terms of transplantation, quite a bit has been done to this point in the eye. So the eye is a good deal ahead, probably 5 or 6 years ahead of the other parts of the brain in terms of stem cell transplantation. And there are currently at least two, and a number more on the way, clinical trials involving transplantation of RPE into the eye for patients with AMD. Now, the caveat here is it's just the retinal pigment epithelium. And as I mentioned before, it's the photoreceptors that actually do the seeing. So if you're just putting in RPE and you're missing both the RPE and the photoreceptors, that's like replacing a carburetor when you're missing a carburetor and pistons, right? So it's not really going to do a tremendous amount. What it's thought to do is to maybe slow the disease process down or help rescue some of the photoreceptors on the edge of the area that's dying and help them be a little more healthy, reduce your scotoma, reduce that central area of blindness that you have. That's about the best that can do, but it's a first step. Myself and Dennis Clegg at the University of California, Santa Barbara, we're trying to combine what we do in the laboratory to create photoreceptor-like cells, combine that with RPE as a next generation approach to try to address the fact that both of those cell types are missing. And even that's a little simplistic because, as I mentioned, there can be scarring, there can be other problems that occur beyond just the photoreceptors and the RPE. So again, we're taking the appropriate steps, it's moving quickly, but for patients who are suffering from these problems, it's never quick enough. So here's a list of some of the trials that are either going on now. I don't expect you to read them, but you can see that there are quite a few. These are trials that are going on now or that are scheduled in the near future for various types of stem cells. None of them are going to cure the disease. None of them are going to get you reading the New York Times again. They're designed basically to set up safety studies to see that the technology is safe. And if there's some improvement in perhaps mobility, a decrease in the central blind spot, that sort of thing. We're located down there at the end. We don't have, certainly we're a long way away from having approval, but we have a good amount of momentum from various companies as well as academic partners to move ahead with the work that we're doing to try to replace photoreceptors in addition to RPE. And we have this capacity here at UW. This is, again, circling back to how great UW is in this whole scheme of things. Not only were they invented here, not only have they been developed here, and people from all over the world come here to get trained and to see what we're doing, but we're also leading the way in taking stem cell technology and bringing it back to the patient. The Weissman Biomanufacturing Facility, which is two floors down from my laboratory, is a state-of-the-art facility that can take human stem cells and, using the protocols we develop, create the cell types that are needed, in this case photoreceptors, and do so in a way that can be put back into a patient. But that takes time, and we're in the middle of doing that right now, but we're making great progress. So to finish up then, what I want to leave you with are just a few points. One is, this all started in, let's see, 1997 or so, and induced pluripotent stem cells, the adult stem cells I've talked about all day today, were invented in 2007. And we're talking about a clinical trial 7 years later. That's unheard of. So this is moving fast, but it's still measured in years, not in months, not in weeks. There's a lot of momentum here. We have to be a little careful that we don't move too fast. We need to make sure that it's safe. There are clinical applications beyond transplantation in terms of discovery of new drugs, testing of drugs, and other approaches that can get to the patient faster. And that doesn't get played up in the media. And the eye itself and the retina really has been way ahead of the field. The rest of the central nerve people who work in the brain and the spinal cord really are watching the retina and following suit. And there's a lot of reasons why that we can get into in terms of accessibility. It's a compartmentalized organ, so if something goes wrong, you can always remove the eye, as we heard. And all the surgical techniques are pretty much straightforward. So there's a lot of challenges that still lie ahead, but we're making good progress. We're farther ahead than I thought we'd ever be, but undoubtedly we'll hit some hurdles along the road, and it's important to keep the expectations reasonable. We're looking to crawl before we walk, before we run. So it's not going to be an overnight, just slam dunk home run. It's going to take a little while for us to refine it. But 10 years ago, even say 5 years ago, I would have had nothing to talk to you about, really, and now we do. So that's very encouraging. So thank you, and I'll take questions.